So, it wasn't you in the end then?
No, I didn't sell your cookies.
Ah, so why'd you run away at the end of the last video?
Well, you know, I always seen in films, chase scenes looked really cool, like fireworks, explosions, running, guns, chases, you know.
So you thought that would be cool. You
didn't
didn't think about the fact that it's a closed room then?
No, that didn't cross my mind until I got over there, 3 paces away, and realised I had nowhere to go, and then you caught me.
Yes, well if it wasn't you, that still begs the question of who stole my cookies. And we've played Cluedo.
Guess who?
Monopoly next?
Yeah, definitely, we should do Monopoly, but first we should solve the crime.
Okay, so someone stole my cookies in the last video, in case you didn't see. We've tried fingerprinting and dental records. There must be something else to narrow it down.
Something else, other than those 2 things that separate people. So other things like appearance, we didn't see them do it. Hair?
Oh, there was a hair found.
There was a hair found at the scene of the crime with a crumb. So it can't have been
crumb.
bald Jim.
No, because he doesn't have any hair.
Yeah.
Can't have been the ginger, because it wasn't a ginger hair.
No, but everyone else has the same hair colour. We're all very boring.
Yeah, so there must be a way of distinguishing between them without using our Sherlock Holmes powers. We must be able to biologically prove.
I reckon we can do that. We can remember how, using our Sherlock Holmes powers, right?
Okay, so Mind Palace.
Right. Oh, mine's messy.
It's fine, mine's got a cleaner. Ah! DNA profiling, so simple. You okay?
I'm getting clear. Alright, got it, okay.
Okay, so DNA profiling, you got that?
Yeah.
Okay, good.
Okay, so to do that, we should know a bit more about DNA.
We do. So what we've done is, at a crime scene, this is a crime scene for stealing cookies, it could be a murder case or whatever, we'll go over the uses later.
Yeah. There'll be some sort of source of DNA, say a hair, and we have the hair. We don't want the whole DNA.
No.
That'd be crazy.
And firstly, what part of the DNA do we want, and then what are we going to do with it? So.
Yeah, so they split into 2 main parts, all of genetic code, and that's stuff that does things, and codes for proteins or what have you, and things that don't appear to code for anything at all.
And the things that code for the proteins and stuff and are known to code for things are called exons, and the things that seem to code for nothing are called introns, and what we're going to use is the latter.
Yeah, we're going to use the introns as the things that's important to us. So the first thing we do is split our DNA into just introns, and we do that using an enzyme.
Yeah. And what we want is a couple of introns, because they're sort of triplets. We want a little repeating unit that's big enough for us to be able to visualise, but not the entire DNA. And we call those short tandem repeats, the parts that we're going to separate into. Restriction endonucleases.
Yeah,
very good word.
It's quite a big word. It doesn't fit on Scrabble.
Not yet.
Not yet. We'll make a big Scrabble.
And so we've got these STRs. We want to get these STRs. They're going to be short bits of DNA.
Yep.
And then we can compare it between what we found at the scene, which would be our hair, and the suspects, which is our studio audience.
So, to do this, we need to get our DNA, but we've only got 1 bit, we'll just split it.
That's not enough,
That's
because
not
what if I pick up this short tandem repeat, and I compare it to, say, Jenny's short tandem repeat?
Oh wait, when you dropped it.
Exactly, that's all gone now, done. Never find it.
What do we do? So you had to replicate it.
We do, and we have a special machine for this, which...
It's called yeah, it's called a polymerase chain reaction, which what undergoes. It's called a PCR machine.
Absolutely. So we've got this PCR, polymerase chain reaction, and we've got a machine. So if you want to get me a Christmas present, I really want one of them, please.
Oh, just get me a Bugatti Veyron, that'd be fine.
I'm sure it would be. I'm sure it would be. So in this polymerase chain reaction, we've got quite a few steps. You've got to know all of them. So it's all about replicating our little hat.
No,
our little
hat.
Shame. Replicating our little short tandem repeats. So there's a few stages.
So, the first stage we have our DNA, and it's in 2 strands.
Yep.
That's fine for now. What else do we have in our test tube?
Well, in the same test tube with it, we're going to have some free nucleotides.
So yeah, free nucleotides, which is just our bases that we'll join later.
Remember, back in protein synthesis, you know about free nucleotides. They line up to make another strand during replication. We also need DNA polymerase. Those are the bases. We need DNA polymerase. That's the enzyme that zips up the hydrogen bonds. We're also going to throw in a few fluorescent primers, just primers we can call them. Now, if we're using this with free nucleotides and DNA polymerase, that implies that this is going to break up and replicate, so we need to break it up.
So to break it up, we need quite a toasty temperature.
And in fact, there's a value given in the book, that you may be asked to quote, 95 degrees Celsius.
And if you do this, it splits into 2 strands.
So this is where we're going to colour code so you can see what's happening to each thing. So are we calling this a second step, or are you just showing what's happening?
Yeah, that's for splitting. We'll call this the first step, and then we'll go into the second step.
Okay. So they split up into their 2 separate things.
Yeah. So once they've split, that's all fine and dandy. Now we need to cool it down for stuff to happen.
To indeed. So we've got.
So we cool it down to another temperature you have to know, that's 55 degrees. And once it's here, then the primer's attached. The primer's attached to each end of our STR.
Yep. So I'm gonna. This little black dot is the primer. It's going to attach to each one.
Yeah. And once it's done that,
It's
that's
ready.
that stage then. So then we heat it up again, but not as much this time, just to 70 degrees. And this is when our DNA polymerase attaches to our primers. They're sort of like markers in this case.
So we've got each of these things.
And they'll sort of attach, yeah, like that, like balls to it. And this will cause all the other 3 nucleotides to join and sort of re-zip 2 new DNAs.
So what we're going to have in the end is 2 DNA strands. The primer's still going to be on each, just like that. And that's going to be 2 complete DNA strands. But that's not
very many, is it, Wayne?
No. So polymerase chain reaction sort of implies
Just
change.
Just keep doing it for ages. So second way round, if we do it one more time, we start with that.
Yep. So now we're going to go over to this 95 degrees again.
Yeah.
We're going to heat it right up. So
We're going to split it all into 4. So we have 4 chains now.
Yeah. So if you want to start drawing that, we're going to have 4 separate ones, as Triv just said. Just like before. But now 2 of them have already got the primer on from before.
So then, when we hit the 55 degrees,
we're going to have 2 reds.
we're going to have more primers that are attached.
And in fact, a primer is going to attach to each of them.
Yeah.
So this is essentially going to have 2. This one's going to have 1. This one's going to have 1. So these 2 are like the first ones. These ones that already had primers are now both going to have
2 primers.
2.
So then it goes again to the next stage, and the primers cause more chains to rejoin, and this happens again. So now we have 4 full chains, but the ones with 2 primers, 1 primer on each end, they can't split again, so that's that bit done.
Absolutely. And then the... so we've got all these. So these 2 are going to be taken off, they're done with. We can have them. And these 2, looking just like the ones before, are going to go around in the chain.
And also keep doing this.
The second temperatures are the same as the first. You go from 95 to 55 to 70.
Sorry, I'm dropping pens everywhere.
Wouldn't do that.
I wouldn't do that either. So then we have much more DNA, but Wayne, what do we do when we have the DNA?
Well,
Now we want to go into test tube, got it?
Yeah. We can't visualise that. That doesn't mean anything. It's not really comparable because you can't lift it up and compare it to, say, Sally's hair. Picking on different people.
Yeah. So instead we do a process called electrophoresis. Now that's pretty snazzy, and for that we're going to need some electrophoresis gel. Now this gel isn't for your hair.
I wouldn't put it in your hair if I were you.
I wouldn't either, that's a bad idea.
And what we're going to do is we're going to have this little
some gel.
Little top thing.
And in our gel we have a trough, and this is where we put our DNA. We've got some DNA in a test tube.
This is our trough.
Yeah, it's just like a dip in the gel. We put it in the dip.
Oh yeah, the gel's going all the way along like this, dotted line. This here is a pipette. You're just going to gently, very small amounts of your sample into that.
So you've done that. That's fine. Nothing has happened yet.
Yeah, it's just going to be stuck in the trough. We need to make it move.
Yeah, DNA won't move by itself. When we do that, we put a potential difference across it. So we use a negative terminal at the side of the DNA. As DNA is negative, it will then move. But each part of the DNA will move in a different amount.
Bigger amounts are going to move less. They're going to be less affected than the really light amounts, as it were, that are going to move further away. And if we look at this
sort of top-down view,
what you're going to have is like it's going to be a rectangle.
A rectangle. You have many troughs, not just one.
Yep. So if we're looking at
this will compare. So we'll put each different type of DNA in each trough, like our suspects and then our real one. We'll compare bands. So what will happen is they'll move a different amount, and these will form bands, which will stay there for a short amount of time. However, if you leave them for too long, they'll move. And there's another thing: you can't always see them.
Yes, at this stage you still can't see them, and there'll be a lot more errors in this. There'll be a lot more little parts to compare, but that's the basic idea. You're going to cover this after it's done in a little nylon sheath.
and nylon sheath will say absorb all it is or keep it how it is.
So you've got this nylon sheath with the absorbed patterns you still can't yet see.
So then, if you say make it fluorescent by
you're putting in a little sort of, this is in a bag or a
Yeah.
box or something.
putting in some fluorescent probes,
Yep. So you can put in probes rather than primers.
then you'll be able to see what it will look like.
Yep.
And it'll be lines.
So there'll be lines where they each stopped along the distance. So some of them will have got a further distance, and so the line will be further on. And you can compare this result to what you'll have done the same thing with a suspect. You'll get a suspect, you'll obtain some of their DNA from a cheek swab or something like that. You could just have got a hair of them as well, whatever. You're going to do the same whole process separately, and at the end compare the profiles, and you'll see if it was the same person that these two pictures, as it were, will line up.
And if they do, they're your man. Because it pretty much always works. Even identical twins have got different DNA profiles.
Yes, because there's general mutations that happen through your lifestyle. They're gonna happen naturally, and so they're going to be different. I think it's about 1 in a billion people, about 1 in a billion cases will actually give a wrong answer here, and that will only be if it's really close family. Very, very rare.
Very rare indeed. So if we use this way, quickly doing a PCR reaction. Alright, done it.
Okay.
Should you, Wayne?
Whoa, what?
You stole your own cookies.
How can I steal my cookies? They're my cookies, right?
Well, that's what I think, Wayne. I think they weren't your cookies. I think they were my cookies. But I thought you were your cookies. So you took your cookies, then realised they were my cookies, and then then swapped them. So I still have my cookies, but you said your cookies were stolen, so they'd be replaced.
I think rather than being a baby, you just think, why'd you put them in such a silly little place? You're so silly, you're silly.